Good morning, dear student. In previous lecture, we already discussed the storage polysaccharide originating from the plant source, that is starch. Starch, it is a combination of amylose and amylopectin. So in this lecture, we are very much interested in the second storage homopolysaccharides. Okay, second storage homopolysaccharides which having the animal origin, that is your glycogen. Okay, so this glycogen, it is your homopolysaccharides. It is a storage homopolysaccharides which having animal origin. Okay, so this glycogen, it is also known as animal starch. So let us discuss some important points regarding to this animal starch. In previous lecture, we already discussed the starch originating from the plants, which consist of amylose and amylopectin. So this glycogen, it is also known as animal starch because its origin is from animal. Okay, so let's discuss the one by one some important points regarding to this glycogen. The first point is indicate that it is a reserve carbohydrates. This glycogen, it is a reserve carbohydrates found in liver and muscles of animals and human beings. So this is the important characteristics of this glycogen, where it is found, that is, it is a reserve carbohydrate found in liver and muscles of animals and human beings. It is also found in plant which do not have a chlorophyll system. Okay, so it is found in plant which do not have a chlorophyll system, means it is not found in green plants. It is not found in green plants, so we can say that it is not found in green plants. So it is found in fungi and yeast. So if we consider, it is found in fungi and yeast, which is not a green plant. Okay, then the third important point regarding to this glycogen is that its molecular weight. According to the different sources, may ranges from 10 raised to 5 to 10 raised to 8. And then each molecule of this glycogen, the next important point is that each molecule of this glycogen contain 5,000 to 10,000 glucose molecule. So each molecule of this glycogen, it consists of 5,000 to 5,000 to 10,000 glucose molecule. Again, if you see the structure of this glycogen, it is a branch structure. So its structure is similar to amylopectin. So we already discussed the structure of amylose and amylopectin. Amylose, it is a linear sequence of this glucose molecule joined together by alpha 1,4 linkage, that is your glycosidic linkage. Okay, and this amylopectin is your branch homopolysaccharide. Okay, in which there is a 1,4 as well as 1,6 linkage is there. And if you see the structure of this glycogen, it is also similar to this amylopectin, means it contains both 1,4 linkage as well as 1,6 linkage. So the next important point is that it is a branch structure. It is a branch structure with straight chain unit. Okay, it is a branch structure with straight chain unit uh, of 12 to 18 alpha D glucopyranols. So again, there is a glucose molecule as a monomer, and it is a 6-membered ring. Therefore, it is considered as a glucopyranols. Okay, so it is a branch structure with straight chain unit contain 12 to 18 alpha D glucopyranols, which is joined by 1,4 linkage, 1,4 glycosidic bond, and with branching. And this straight change, not only there is a straight change, but also the straight chains are joined with another chain by 1,6 linkage, so there is a branching with another linear sequence of this polysaccharides. Okay, of this polysaccharides, this is called 1,6 linkage. Okay, 1,6 linkage, that is the alcoholic group of the sixth carbon. Okay, or the sixth carbon, which is alcoholic group, it will interact with your carbon number 1 of first monomeric chain. okay, a linear chain of this glucose molecule. With elimination of one water molecule, it will forms 1,6 linkage. So each molecule, it has a branch structure with a stretch chain unit of 12 to 18 alpha D glucopyranones joined by 1,4 linkage, and with branching. So there is a branching also because its structure is similar to this amylopectin. Structure is similar to this amylopectin. Okay, with branching, that is 1,6 linkage. If you see, the next important point regarding this glycogen is it is a non-reducing sugar. It is a non-reducing sugar, means it does not have a free aldehyde group. So you should keep in mind, we already discussed what is reducing and what is non-reducing sugar. Reducing sugar which having a free aldehydic or ketonic group, which is undergo reduction, which is undergo oxidation, not reduction. It will react with your oxidizing agent, and the conversion of this aldehyde group into acidic group. So there is oxidation of this free aldehyde group and convert into acidic form. So we already discussed the reactions of this reducing sugar which having a free aldehyde group, that is reaction with Tollens reagent, reaction with filling solution, reaction with Benedict solution. So they all are act as oxidizing agent. So it will oxidize the aldehyde group into acid and itself it reduce. So we already know what is the reducing and non-reducing sugar. So the reducing sugar, which shows positive test with your Tollens reagent, filling solution and Benedict solution, and those sugar or carbohydrate which does not give positive test with your Tollens reagent, okay, filling solution or Benedict solution, they are considered as a non-reducing sugar. So this glycogen is your non-reducing sugar. It is readily soluble in water. It is readily soluble in water, and again it will gives the red color with I2. So we already discussed that in the starch which having the plant origin, it having two constituent, one is amylose and second is amylopectin. This amylose gives blue color with iodine solution. So you should keep in mind the different colors of this polysaccharides. That is, amylose gives blue color with iodine solution, then amylopectin gives violet color. It will gives violet color with this iodine solution, and this glycogen, it will gives a red color with I2 solution. Again, if we see the structure is similar to this amylopectin. So amylopectin, we already discussed this amylopectin. It is a branch homopolysaccharides. It is a constituent of starch. Okay, so this is not a linear sequence of this. There is not a linear sequence only, so there is a branching also. Okay, so it consists of 1,4 linkage as well as 1,6 linkage. So whenever there is a 1,6 linkage, then there is a branch, then there is a branch. So its structure is similar to this amylopectin. Its structure is similar to this amylopectin. And if you see this glycogen, it is more highly branched than amylopectin. No doubt its structure is similar to this amylopectin, but it is more branch. Okay, it more or highly branched, we can say that it is highly branched than amylopectin. Highly branched than amylopectin. So you should keep in mind, no doubt the structure is similar to that of amylopectin, but it is highly branched, than amylopectin. Okay, and its branch point is up to 8 to 10 units. Its branch point is up to 8 to 10 glucose unit. So in previous lecture we already discussed the amylopectin. It In amylopectin, the branch point is 20 to 25 glucose unit. So in this case, the branching at 8 to 10 glucose unit. Again, the glycogen which is on hydrolysis gives glucose, maltose, and some small amount of dextrin. Okay, so this is all about the glycogen which having the animal origin. It is also known as animal starch, and this both starch and this glycogen, okay, the starch which having the plant origin and the glycogen having animal origin, both are storage homopolysaccharides. So we already discussed what is the difference between homo and heteropolysaccharides. Homopolysaccharides These are the polysaccharides which is on hydrolysis gives single type of monosaccharide molecule, and therefore they are considered as a homopolysaccharides. Okay, and the heteropolysaccharides which is on hydrolysis gives the mixture of different constituent. Okay, and some derived products, they are considered as the heteropolysaccharides. So this is all about the storage polysaccharide. We discussed the two important storage polysaccharides, which having plant origin, that is your starch, and which having animal origin, that is your glycogen. In the next lecture, we will discuss the structural polysaccharides, structural homopolysaccharide, that is your cellulose. Thank you.